Students, let's discuss meiosis, that is animal meiosis, which is a type of cell division seen during gamete formation. So meiosis is the phenomenon which occurs in any life cycle that involves the process of sexual reproduction. The production of offspring by sexual reproduction involves the fusion of two gametes, and the gametes are formed as male and female reproductive organs by meiotic cell division. So meiosis is a special form of cell division which reduces the chromosome number in such a way that each daughter cell receives only one set of each kind of chromosome. So this is called a reductional division. So meiosis is called as a reductional division. So why? Because the chromosome is reduced to half. So what are the essential features of meiosis? It undergoes two successive cycles, meiosis 1 as well as meiosis 2. After the replication of parental chromosome, meiosis 1 takes place in order to produce identical sister chromatids at S phase, and meiosis takes about days to get completed instead of hours or minutes that are needed for mitosis. Pairing of homologous chromosome and recombination takes place during meiosis. Finally, after the second meiotic division, 4 haploid cells are formed. So you can see here, this the 4 haploid cells are formed from the one cell. That's called reductional division. Now meiosis 1. Now meiosis 1 is a very long phase and it includes prophase, metaphase, anaphase and telophase, but prophase is of longer duration, and in that prophase is again having many 5 stages: leptotin, zygotin, patchotin, diplotin, diakinesis. So up to this is a prophase 1. So it involves leptotin, zygotin, patchotin, diplotin and diakinesis. So you can see clearly in the figure, leptotin where obviously chromosomes will be looking clearly from the chromatin fibers, and here centrioles get divided, no disappearance of nucleolus and no disappearance of nuclear membrane. Now in leptotin phase, nucleolus is there, cytoplasm is there, nucleus is there, chromosomes are there, centromere is also there, and centrosome get divided into centriole, and centrioles are moving towards the opposite poles. In zygotic phase, there is the homologous chromosomes, that means this which are looking same shape and size, like metacentric, then metacentric, submetacentric, then submetacentric. So shape and size may the homologous chromosomes, they look same and they arranged in pair, and this pairing is called as synapsis. So here this zygotic phase, homologous chromosomes occur in pairs, and that's called synapsis. Now synapse chromosomes, you can see patchotin stage. Now the synapse chromosome, the chromatids come closer to each other, and then patchotin stage, this crossing over start, and a crossing over means what? The exchange of chromosomal material. And now in diploid stage, that crossing over ends. So here already exchange of chromosomal material done, but the point of contact, that is chiasmata, remain the same, it's not separated. So in diakinesis phase, they remain attached to each other. Once the crossing over is over, but they still attached and they look like loops. And these looped chromosomes, that's what we say is a tetrad, because of 4 chromatids are there. So this is a tetrad where 4 chromatids are there, and it is also called a bivalent. And these get arranged in the metaphase plate in the same way as in mitosis. And in the metaphase plate, when they are arranged, then they are divided into 2 univalent or diad. You can see these dyads are moving towards the opposite pole through the spindle fibers. Now spindle fibers are the proteins, and here in telophase 1 they have already reached, and then immediately two daughter cells are formed, and then these 2 daughter cells again the prophase 2 start, that is meiosis 2 starts, and meiosis 2 also contain prophase 2, metaphase 2, anaphase 2 and telophase 2, which is simple type of mitosis. So here you can see again the centrosome divides into 2 centrioles, spindle fibers are formed, and the dyads are arranged, and now finally the dyads are splitted up into 2 chromosomes, and each pole is receiving half of the chromosomes, and finally 4 daughter cells are formed from the one cell, and this all 4 daughter cells, each one is having haploid nucleus, but the original nucleus bearing cell is the 2n1, that is a diploid one. So 4 daughter cells are formed which are haploid in nature, and this happens during gamete formation. And here is the special thing happens, that is crossing over, that is shuffling of chromosomes, shuffling of genetic material, which introduces variation. So what is the significance? So the significance of meiosis is maintains the same chromosome number, that is reduced to half. So significance is gamete formation takes place, it's a reductional division, and here it introduces variation and shuffling of chromosome. That's why we all are different from each other. So just go through NCERT, read NCERT thoroughly and join the class. Thank you very much.